So the first reaction is done by either hexokinase or glucokinase. You're going to hear me talk primarily about hexokinase. We'll discuss the glucokinase momentarily. So the first reaction is we're going to phosphorylate glucose. This is going to be a priming reaction. So this is actually going to utilize and consume ATP. We'll get that back later, but this priming reaction is really important for a variety of different reasons. For one, that is going to make this process spontaneous, so that ATP utilization is going to be a coupled reaction that's going to allow this reaction to happen spontaneously and move forward. So below what you can see is the reaction. So we start off with glucose plus an ATP, and our enzyme is then going to allow this to then be phosphorylated on that 6th carbon, which is why you then have the product being glucose 6-phosphate. As we go through all of these steps, it really helps if you will actually go ahead and take a scrap piece of paper and draw these out, because all of these names relate back to the chemical of what's actually happening here. So the more you're familiar with these structures, the better this entire thing will go. So with those thermodynamics, so again, this is a coupled reaction. So if we were talking about doing this without ATP, if we were going to try to phosphorylate this with just inorganic phosphate, that's actually unfavorable. So it is that hydrolysis of the ATP that's going to let this drive this reaction forward and be a spontaneous reaction. The other thing that actually helps with this reaction is in the cell, the product, your glucose 6-phosphate, is about 100-fold lower than the concentration of glucose. And the ADP is usually about 100-fold lower than ATP. So having both of those products be in a lower concentration than the reactants also helps drive this reaction forward. So that remembering that we used to have this equation we talked about in thermodynamics, where we could talk about the delta G value and what was actually happening right now in whatever conditions we had, and we were talking about the fact that we would use Q, which is that products over reactants. So it's been a while since we looked at that, so just that reminder that those concentrations do play a role in whether or not a reaction moves forward. Under cellular conditions, the actual delta G value is going to be this negative 33.9, which again, highly favorable, is going to drive our reaction forward. So not only is this ATP important for driving the reaction forward thermodynamically, it's important for other reasons as well. So one, this is going to basically set us up for glycolysis to release energy later. So it is what's called this priming reaction, in that it's going to give us really high energy intermediates, so that glucose 6-phosphate's got high energy to it, and so we're going to be able to release that energy later. So even though we're putting energy in, it's actually going to help us to release more energy down the road. Another one is that this is also going to introduce a negative charge to that glucose molecule. So going to glucose 6-phosphate, remember phosphate's got a negative charge on it, which means your glucose is not going to be able to cross that membrane of the cell by just passive diffusion. So your glucose can get in by passive diffusion. It can go ahead and get through that lipid membrane. It's a relatively small molecule, but it can't get back out again now that it has the phosphate on it. So it's a way for the cell to spend a little bit of energy, but to hold on to it. So it's a way for the cell to hold on to the energy it's got. The last one then is that rapid phosphorylation of glucose is going to help the cellular glucose concentration stay low. So when we're talking about this notion of glucose coming into the cell, this is just going across the lipid membrane. The thing is that the more glucose that is outside the cell, the more that will want to come in. So you have that passive diffusion. Well, if the amount of glucose inside the cell and the amount of glucose outside the cell are the same, you're not going to get any more diffusion in. So it's useful to have the amount inside the cell be lower so that you keep having more glucose come in over time. So with that in mind, what are you going to do with all this extra glucose? Well, if we go ahead and phosphorylate it, glucose 6-phosphate's a different molecule. So you're not going to have that equilibrium between the glucoses. So we're going to get rid of some of the glucose inside of the cell to glucose 6-phosphate. And that's going to help the cell continue to keep more glucose coming in. And the other thing here just to mention is that, again, the glucose 6-phosphate since its charge won't be able to leave. So it's to keep what you've got, helps with the equilibrium, all of these things. Glucose 6-phosphate's also common in several metabolic pathways. So while glucose is really useful, because that glucose 6-phosphate is that kind of first step as far as like, this is mine, says the cell. Therefore, it's actually utilized in a couple of different things. So we can actually use glucose 6-phosphate either directly in the pentose phosphate pathway, or you can actually get that changed to glucose 1-phosphate, which is then used to create glycogen, or can then be moved into glucuronate, which is used for carbohydrate synthesis. And even as we move down the line, glucose 6-phosphate will then go to fructose 6-phosphate, which again can be utilized to glucosamine 6-phosphate to then carbohydrate synthesis. So even looking at a small snapshot of cell metabolism, glucose 6-phosphate starts us down where we've got a lot of different potential chain reactions that can happen. Hexokinase is the enzyme that does this reaction, and what's really neat about it is that it's got a beautiful induced fit to it. So when we look at this, is that the mammalian hexokinase has both an N-terminal and a C-terminal domain. And then when we look at these, it's got a really cool induced fit mechanism. So I realized that B is actually the glucokinase. These two enzymes are very similar. But what you can see is you basically have almost like this like clamshell notion where like the bottom part comes up to the top part. And so it's got this really lovely section that actually moves with it. So it's not a static enzyme. It has a lot of movement in it to get that substrate to actually come together. Being able to actually phosphorylate glucose, that reaction actually is done mostly by proximity. So that induced fit becomes really important because it's mostly the proximity of the correct part of the glucose molecule to the ATP that allows that to get phosphorylated. So this whole idea of glucokinase and hexokinase. So again, they are actually very similar enzymes. They do the same reaction, and they actually have very similar structures to them. But hexokinase has a really low Km value, so it's got a much tighter binding to it, and it also acts in most tissues. Glucokinase, on the other hand, is predominantly found in the liver as a much higher Km, so about a 10 millimolar Km. Glucokinase you really only see in the liver when dietary glucose levels are high. So glucokinase also isn't something you actually find at all times, even when it's in the liver. That induction of making a lot of glucokinase is controlled by insulin. So whenever you have a lot of glucose, then it can be phosphorylated in the liver by glucokinase and converted to glycogen. So glucokinase is almost kind of like this backup enzyme that when you have it, it's trying to help deal with this influx of a huge amount of glucose. So then when you talk about patients who have diabetes, this is going to be providing insufficient insulin. So because patients may have low levels of insulin, they're also going to have low levels of liver glucokinase. So they don't have that backup system to help deal with all this extra glucose. So they're only dealing with mainly hexokinase and not that kind of backup to help them out. And so this is going to lead them to not be able to tolerate high levels of glucose in the diet because their bodies just aren't processing it very well. And because of the lack of your glucokinase, this is also going to lead it to not produce very much liver glycogen either. So not only are they not processing what they've got, they're also not storing the extra very well. What should normally happen is when glucose is high, insulin should stimulate the uptake of glucose in the cells, as well as synthesis of glycogen and other anabolic reactions. Like, hey, we have all this extra sugar, let's store stuff, let's make stuff because we have so much extra. But in diabetes, when glucose is high, it's high in part because it doesn't have a lot of the glucokinase to help take care of it. So again, diabetes, they're not going to be producing that glycogen at a very high rate. So they're not actually doing those storage mechanisms like they should be able to. So they're still actually usually working with things like glycogen breakdown and potentially gluconeogenesis, which actually makes more glucose. Because your glucose uptake's impaired, your diabetics are going to have a high glucose in the bloodstream, but are also going to be characterized by biochemical starvation. So again, with diabetes, you're not taking care of sugar the way you're supposed to. So it's not just that there's too much extra and you can't handle the extra, but your body's also not actually processing a normal amount of extra. So it's not processing the normal amount to storage as should normally happen. There are two kinds. So insulin dependent type 1 diabetes mellitus is characterized by an autoimmune destruction of insulin producing cells in the pancreas, beginning either by age 20 or earlier. So this is usually when you talk about either childhood diabetes, or I don't know if you've had any friends that have actually come down with type 1 diabetes while they're in college. I actually had a couple of friends that had that. It was kind of like, I thought only little kids had type 1 diabetes. And no, it can actually come on later in your late teens, early 20s. Non-insulin dependent or type 2 diabetes, this is going to occur usually in middle age, and it's usually associated with diabetes. So insulin is made normally, but your cells basically don't receive the signal like they're supposed to. The exact mechanism is not super well understood, but what's good is that they can often be treated with basically a dietary and lifestyle changes. Our second reaction, phosphoglucoisomerase. So this sounds really fancy, but if you break it down, realize what we're talking about with this enzyme is phosphogluco, so a phosphorylated version of glucose as our substrate, and then an isomerase. So we're going to have some sort of isomerase reaction on a phosphorylated version of glucose. Well, since our first reaction, phosphorylated glucose, and we currently have glucose 6-phosphate to deal with, should not be a big surprise that that's going to be our substrate in the second reaction. So what we're doing here is we're having a phosphoglucoisomerase, which is going to transfer this from glucose 6-phosphate to fructose 6-phosphate. Why on earth are we doing this? What this is going to help with is that the next step, which is going to be phosphorylating at the C1 position, would have actually been really difficult on glucose for that hemiacetal. But it's a lot easier for a primary hydroxyl group. So getting this next step, our third reaction to go, is going to be a lot better for fructose 6-phosphate than it is for glucose 6-phosphate. This is also actually going to help to activate the C3 carbon for cleavage later on down the line by the aldose reaction. We're going to take a closer look at the intermediate. We'll have an indiol intermediate when we look at the mechanism. This reaction actually has a really low delta G value. So this delta G value is about negative 2.9 kilojoules per mole. So that's near equilibrium. So this is one of those reactions that actually is readily reversible. So phosphoglucoisomerase, if there's a lot of glucose 6-phosphate, it'll go ahead and move forward into fructose 6-phosphate without a problem. But if that fructose 6-phosphate backs up and you have a lot of fructose 6-phosphate present, it has no problem going back the other direction to glucose 6-phosphate. So just to remind you that some of the differences that are happening here, so since we're going from glucose to fructose, we are essentially going from an aldose sugar to a ketose sugar. So that's the main thing that's happening here, is with the placement of that carbonyl carbon. Because we're dealing with the ring form of both of these, it's sometimes not as easy to see what's actually happening here, but that's part of what's happening as we're changing the type of sugar this is. There's a picture of phosphoglucoisomerase. Look at all the pretty alpha helices. And fructose here is shown bound in blue. For our mechanism, the main thing I want you to realize is this is a good time to pause the video or go back and look at the PowerPoints and take a look at what's actually happening. But the main thing I want you to pay attention to is as we're looking at this, here you see this circle of E, that's for our enzyme, and then the B with the 2 dots is for a base. So what we have is we're actually going to have a fair amount of acid-base chemistry happening. So what's happening is that enzyme is abstracting a proton, which then is going to help with the ring opening. So in this first step, you basically have a ring opening mechanism done with some acid-base chemistry. Then from there, we're going to continue with more abstractions of protons, though now onto the second carbon, which is going to allow, as we continue down into the second step, for that indiol intermediate. So now we have that double bond in between the 2 carbons, which is then going to allow us to then add a proton back across the double bond, which will then be able to, again, change that aldose sugar to a ketose sugar. So then as we proceed into step 3, our ring is technically still open here. So going from out of the indiol intermediate over then to step 3, essentially what we're doing is we're using more acid-base chemistry, more abstraction of protons, though, to just go ahead and close the ring. And then once our ring is closed, we're finally left with the more familiar picture of fructose 6-phosphate.